Hello and good morning, everyone. This is your instructor, Dr. Monica Gullian-Nass. We will be talking about DNA damage and repair today for our Friday's lecture. We have been talking about DNA damage and repair for last two sets, our last two lectures. Today's topic I will talk a little bit about DNA damage because we have talked about some of the ways DNA can be damaged, but we will talk a little bit more today. And then I will also cover DNA repair. Excision repair and direct repair are the two main methods of DNA repair. We will discuss both of those. Then I will talk about site-directed mutagenesis, irradiation, and also post-applicative repair today. Okay. So this we already covered in Wednesday's lesson, but I just wanted to make sure that we are all on the same page. And remember that DNA damage not only caused during replication due to an insertion of a wrong nucleotide, but it can also happen due to number of different ways. We talked about how the spontaneous water molecules can damage DNA in a cell. This can also occur by alkalination or oxidation and radiation of DNA. And we talked about EMS, that's the most commonly used alkylating agent in the labs to cause random mutations in DNA. Methylation of guanine or formation of methyloguanine also affects the DNA structure, the sequence of DNA molecule, because it pairs with T, and that will lead to from change of GC to AT. So if these damages aren't repaired, then that becomes a mutation eventually. So today we are going to talk about oxidative damage. Now, oxidative damage is when reactive oxygen species which can be generated by ionizing radiations or chemicals that generate free radicals. The reactive oxygen species can change the pairing of these different bases as well. As you all probably know already what free radicals are, these are highly reactive molecules. These are usually short-lived, but even for their short life, they can affect the DNA molecule, both the bases as well as the sugar phosphate backbone. The most frequent oxidative DNA damage that happens is formation of 8-oxo-DG or 8-oxo-deoxyguanosine. This 8-oxo-deoxyguanosine, you can see here, this is guanine molecule. This guanine molecule can convert into this 8-oxoguanosine. And that 8-oxoguanosine is usually formed by the attack of hydroxyl radicals. And this 8-oxo-DG will prefer to bind to A instead of C, that it should bind to. The guanine should bind to a cytosine, but instead of binding to a C, DG binds to A, and it will give rise to a GC to TA transversion. This 8-oxo-DG induced by DNA damaging agent is usually repaired very rapidly in the system. Some of the experiments have shown that when mice are subjected to ionizing radiation, there was an tenfold increase in 8-oxo-DG levels in the liver of these mice. Those are subjected to ionizing radiation. However, most of this 8-oxo-DG was removed within few minutes, and the half-life was 11 minutes. So within this time period, most of this 8-DG was removed from the mice liver, and that what that suggests is the cells have this capability. Cells already are aware of the formation of this 8-oxo-deoxyguanosine, that it's rapidly removed to form guanine. Yeah, because these increased level of 8-oxo-DG or 8-oxo-deoxyguanosine, they serve as a marker of oxidative stress, because when higher level of oxidative stress lead to higher level of 8-oxo-DG formation as well. although most of this 8-oxo-DG will be removed in a healthy cell. If something happened in a healthy organism and the level of 8-oxo-DG is high, that will be removed. But in unhealthy cells, this could be a problem, and that's the case of the case of oxidative stress. So if the cells is under stress, then these levels can be high, and they stay high, and these are a biomarker of oxidative stress. Usually clinicians use these levels to see whether the tissue is under oxidative stress. These are frequently, higher level of OXO-DG found frequently during carcinogenesis events. That means the chemicals that can lead to cancer formation, or which are carcinogenic. If the tissue is affected by these compounds, these molecules, those tissue might have higher level of 8-oxo-DG, similar to the example I gave you earlier. When the mice were subjected to ionizing radiation, they had high levels of 8-oxo-DG, which shows that they had a higher oxidative stress at that given time. However, since these mice were healthy, the cells were able to improve, able to overcome that stress. Another way of which very common form of DNA damage is pyrimidine dimer formation. Pyrimidines, cytosine and thiamines, are two pyrimidines. Right? These pyrimidines can make dimers. This can be a CT dimer or a TT dimer. Most commonly, it's a TT. Two thiamines make a dimer. Making a dimer is more common than a CT dimer, but it could be either. This sort of damage is usually caused by UV light, but there are multiple other ways as well when the thymine dimer can be formed. But UV light is one of the most common reason for this thymine dimer to form, because UV radiations are lower wavelength. These wavelength about 260 nanometer. These are absorbed by the bases very strongly, and they result in this photochemical fusion of two pyrimidines that occupy adjacent positions. So that could be, again, it could be CT or 2Ts. The cyclobutane ring that forms between these two pyrimidines is especially thiamines, as I said. Now, this thiamine dimer, once they made this cyclobutane ring, they cannot form hydrogen bond with the bases on the opposite side. So the bases on the opposite side are just hanging there, so they're just loose, and it will block the DNA replication. However, I will show you later in post-applicative damage repair that when the damage is too high, for example, the tissue, the organism has been hit by these UV radiations. If the damage is high, sometimes the replication machinery just skips this portion and deals with it later and moves on with the replication fork. But most of the time, if the damage is low, then replication fork will halt here, will stop here, and this need to be repaired first in order for replication to move along. Strand breaks, that means the sugar phosphate bond breaking of the DNA, breaking in the DNA is another damage that can be caused to DNA. Most of the time it is caused by free radicals. Gamma and X radiations, they have very high energy. They can ionize the solvents to form these free radicals, and these free radicals then can damage the sugar phosphate bond of DNA. These free radicals, as I said, these are highly reactive, short-lived molecules. These preferentially attack the DNA backbone because the backbone is most exposed part of the DNA. Higher radiation levels are more likely to cause double-stranded breaks, and these double-stranded breaks that we have been talking about in the class for multiple times these are really difficult to repair because when it's a single-stranded break, there is a template strand that can be used to fix the error, but when it's a double-stranded break, there is no template strand per se to fix that error. Okay. So this just talks a little bit more about what reactive oxygen species are and what a free radical is. So I'm not going to go too much into this, but you can read it later. So far we talked about multiple different ways DNA can be damaged. Deamination is one of the ways DNA can be damaged, or DNA can be changed. For example, here, deamination of 5-methylcytosine converts it into thiamine. We talked about DNA damage caused by alkylation, oxidation, and radiation. So all of these different ways can affect the DNA molecule as well. Pyrimidine dimer formation, for example, thymine dimer formation, most common sort of pyrimidine dimer formation that happen due to UV radiations most of the time. That's again as a very hard to repair damage to DNA. Strand breaks as the cleavage of sugar phosphate backbone by free radicals. So all of these different ways DNA can be damaged. So this is an example. We talked about alkylation can happen at either N or O. Oxidation can happen to the base, or deamination of the base. Any of these different modifications will change the DNA molecule, or will damage the DNA molecule. You all have probably heard about the carcinogenic activity of or carcinogenic potential of ethidium bromide. Ethidium bromide that's commonly used in the labs because ethidium bromide, what it does, it stacks within the bases of DNA, and then when we check the DNA under the UV radiation, this ethidium bromide will show up, so we know where the DNA is because of the color intensity of ethidium bromide. So we use it heavily in the labs to visualize DNA molecules. However, it's a carcinogenic compound, so it has to be handled properly as well. And how it works is a flat molecule. Here you can see this is ethidium bromide. Ethidium bromide, it's because it's a flat molecule, it can insert itself between these the bases here in the DNA. So this is an intercalated molecule of ethidium bromide. Now these are nitrogen bases, but how, when it's intercalate itself or insert itself between these base pairs, it moves the base pair. Forms this gap. That's in this gap it sits. So it's typically the distance between two base pair is doubled, and that causes the DNA polymerase to insert extra nucleotide, or DNA polymerase to skip a nucleotide. So it can lead to addition or deletion of a base pair, and that will lead to frameshift mutations, and that's why it's carcinogenic. Okay. So we talked about DNA damage repair for mismatch repairing or replication errors. So this is the sort of repair we have already talked about. This is called replication errors repair, and we talked about how it's in E. coli, the enzymes that are used are MUT-S, MUT-L, and MUT-H. The homologous or orthologous to these enzymes in eukaryotes are MSH, MLH, and PMS in humans, and multiple others as well. This mismatch is repaired during the replication or right after the replication of the DNA molecule. Most of the time in E. coli, it's after the fact. In eukaryotes, it's more as the replication is moving along. This mismatch repair is also going on at the same time to make sure that replication is not leading to any sort of errors, or it can be repaired immediately if there is an error. Number of different type of DNA damage repairs, because there are a number of different type of DNA damages post-replicative. So we will be talking more about these type of DNA damages today. Are photoreactivation, base excision repair, nucleotide excision repair. These are both part of the excision repair, DNA damage repair, and then double-stranded break repair. We'll talk about all these today. So we talked a lot about DNA damage, but how this damage can be repaired. So different DNA damages require a number of different mechanisms to be repaired as well. The repair usually involves either removing the damaged DNA or replacing it with something new, or reversing the damage. So either one of these mechanisms will be used in order for DNA to be repaired. The two methods to repair bulky damage. When I say bulky, that means it's more than one nucleotide is usually involved, or it's a longer stretch that need to be repaired in DNA. Two of the major methods of DNA repair are excision repair and direct DNA repair. Excision repair, we will talk first about excision repair in prokaryotes. In excision repair, damaged DNA is removed instead of repairing, so it's removed and new DNA is formed in that area, in that region. So the intact, undamaged complementary strand of DNA will act as a template to replace the damaged DNA. There are two major types of excision repair, base excision and nucleotide excision. So we will talk about both of those now. In base excision, and think about it, when we say base, that means there's just one part of your nucleotide. So nucleotide consists of three different parts. In nucleotide, there is a sugar, a deoxyribose, there is a phosphate, 3 phosphates, and a base. So it could be adenine, guanine, cytosine, and thymine. When we're talking about base excision, we're talking about that the base is damaged and need to be repaired. The enzyme DNA glycosylase breaks the bond between damaged base and its sugar. It results in an apurinic or apyrimidinic or AP site or abasic site on the strand. Remember on Wednesday we talked about these abasic site. That means a base has been removed. So there's no base in the nucleotide, but sugar and phosphate is still there. So that's what DNA glycosylase does, is it removes the base from the site of the strand. Humans have 11 DNA glycosylase isozymes for different damage forms. And when I say isozymes, that means these are different isoforms of the same enzyme. These apyrimidinic or purinic endonucleases, these are different enzymes. They make a nick at 5' to the site, and this allow the DNA pole 1 to remove this AP site, and then strand repair is finished with the DNA ligase, which seals and nicks. In nucleotide excision, which works on rather more bulky damage that includes thymine dimers, the mechanism is a little different. In E. coli, UVR A, B, and C endonucleases, these are called excision nucleases or exonucleases. These recognize the damaged bases and cut at sites that flank the damaged strand. Most of the time, it will remove 12 to 13 nucleotide, leading to the damaged nucleotide, and this resulting single-stranded segment of the healthy strand. So we have double-stranded DNA. We removed 12 to 13 bases on the damaged strand, but the healthy strand is still there. That healthy strand will act as a template to fill in the reaction using DNA pole 1 and ligase in E. coli. So see here we are talking about DNA pole 1 because pole 1 is more of the polymerase that is involved in DNA damage repair in E. coli. So here both of these excision repair mechanisms together, base excision repair and nucleotide excision repair. Here in base excision repair, there is a deaminated C instead of a C. So deaminated C, when C gets deaminated, it converts into U, right? Uracil. So instead of a C here, it's a U. This U need to be removed and replaced with the C. This is hydrogen bonded base pair right here, all of these. This uracil will, because it's an uncommon nucleotide for DNA, it does not exist, so the cell knows this is the wrong nucleotide. It uses uracil DNA glycosylase. This uracil DNA glycosylase will make a cut, we will break it from the sugar bond. So sugar phosphate bond is still there, but the U is missing, so that removes the base, excise the base. So this is a DNA helix with missing base. Now the AP endonuclease and phosphodiesterase, these remove the sugar phosphate from that site as well, and they will bring a new nucleotide together. The DNA pole will bring the new nucleotide and adds this nucleotide, and ligase will seal the nick. So this is now complete. So this is not the bulky damage here per se, so it's more of a smaller damage, just a single base damage at a given time that is being repaired by base excision repair. In nucleotide excision repair, this can be bulky damage, for example, pyrimidine dimer here, a dimer between two pyrimidines, a cytosine and a thiamine. This type of this bulkier damage will lead to nucleotide excision repair. Again, these are hydrogen bonded base pair, but these make this cyclohexane ring, and that's a need that cannot that need to be corrected in order for replication to occur. And next, nuclease will come and make a cut. So here you can see this nuclease made a cut here and made a cut here. These are endonucleases. These endonucleases, they've made two cuts to remove this 12 to 13 base pair segment. This 12 to 13 base pair segment, including thiamine dimer, has been now removed. And this once it's removed, then this healthy DNA strand will act as a template, and DNA polymerase will fill this gap, and the DNA ligase will then fill the nicks on both sides, and then now the DNA is repaired. So that was the excision repair, both the different types of excision repairs. Now we're going to move on to direct repair. Some of the damages in DNA can be repaired directly instead of cutting the DNA sequence and fixing it like with a new strand. This can be most usually the thiamine dimers, because thiamine dimers happen very often in the cells, and these need to be repaired in multiple different ways. So these they're different organisms have different ways, cells have different ways to deal with this sort of modification. Photoreactivation or light repair is used to fix thiamine dimers in E. coli and in some eukaryotes, but humans don't use this. Human cells don't use photoreactivation. What happens in photoreactivation is again, it's usually occurred by UV radiation, but the energy from light, from visible light, can help catalyze the reaction to in order to fix the damage. So DNA photolyases are the enzymes that recognize these thiamine dimers, and they break the cyclobutane ring using the energy from light. So here's the example. This is a cracked DNA molecule. UV light hit and made these thiamine dimers, leads to these pyrimidine dimers, because there were two thiamines here. DNA photolyase in the dark, for DNA photolyase will come and bind to this dimer, this pyrimidine dimer, and when the light hits it, in presence of this visible light, using this energy from light, this photolyase will open these thiamine dimers, or will remove this cyclobutane ring formation, and then photolyase will remove itself from there as well. And now these two thiamines are separate, and instead of cutting and fixing it, this can be directly repaired in E. coli. Dealkylation is direct removal of methyl group from methylated methyl O6-methylguanine. And what happens in dealkylation is instead of doing a base excision or nucleotide excision, this methyl group, the enzyme can remove the methyl group from methylated guanine, and then the guanine is fine after that. Methyltransferases are the enzymes that can remove the methyl groups. These are called suicide enzyme because they can recognize methylated guanine and transfer the methyl group to its own cysteine residue. But once once the methyl group attaches to its cysteine residue, it inhibits its further activity. So the enzyme is dead at this point. So here, this example, this is a methylated guanine. This methylated guanine methyltransferase enzyme, this has this cysteine residues have this sulfohydroxyl group. This sulfohydroxyl group will take this methyl group from here. This methyl group will bind to the sulfohydroxyl, and here it has removed the methyl from guanine, so that way the guanine is correct. Now the DNA is repaired, but this enzyme is no more activated, so this will be dead. If there is another damage, there need to be a new enzyme for this. That's why it's called suicide enzyme. There are many ways the researchers use these different methods of causing DNA damage to understand the function of the genes, or repairing the damage that has happened, to understand how these different mechanisms work. So most of the time, how to know how something works is by changing it and seeing what happened to its activity. Say, for example, if we knock out methyltransferases, what happened to the cell? So the if there is no methyltransferases, it will not be able to correct this methylguanine or methylated guanine into regular guanine, for instance. Similarly, if you knock out photolyase in E. coli, what will happen if you hit the E. coli cells with UV radiation? So that kind of experiment tell us or show us what is the function of that enzyme and what it might be doing. Now, a lot of early experiments were carried out with random mutagenesis. So for example, EMS or radiation. Radiation have been heavily used in insect control as well, by making the sterile insects, when we will talk about this later in the lecture. Once you use random mutagenesis method, either way, whether it's a chemical or radiation, and then we follow different organisms, because different organisms will have different phenotype depending where the error has happened in their cell, and that helps us learn the gene functions as well. It also helps learn DNA repair, mechanisms of DNA repair. So we talked about excision repair in prokaryotes. Now, how excision repair works in eukaryotes. The general principle, general mechanism is quite similar in both eukaryotes and prokaryotes. The enzymes, the proteins that are used in eukaryotes are called XP proteins, and XP stands for xeroderma pigmentosum, because that's the disease. It happens when it's mutated. And a lot of gene names are depends on what the functions they have. In eukaryotes, a specialized protein, which is called XPC, it recognizes the damage. This XPC, once it's recognized the damage, then it will recruit other proteins. Some of these proteins are XPA, which is an helicase. It helps unwind the DNA. And an endonuclease, which is XPG. So all these different proteins, they bind to DNA molecules wherever there is a damage. So for example, here this looks like a thymine dimer, or a pyrimidine dimer. This pyrimidine dimer is recognized by some of these XP proteins, and these XP proteins come and bind to these. This is XPC here. This recognizes the damage. It's XPE and DDB1, the double-stranded DNA binding proteins. They come and bind to these molecules. These will recruit, or these will tell that there is a damage. So XPC comes and bind to this place where the damage has happened. Once XPC binds there, then it recruits multiple different proteins, including these XPA, XPG, XPB, XPD, and this start, the DNA is opened here. This once the DNA is opened, then it will make a break and remove the portion of the DNA that's broken, and a new using the correct strand, the new DNA strand will be formed. This, as it says, the single-stranded region is now filled by DNA pole and ligase, because it makes this cut. The XPG makes the cut in wherever it finds the damage. And again, this is going to be a multiple nucleotide removal, not just a single removal, since it's considered as a bulky damage. In case when if the DNA damage is discovered by the RNA polymerase before the helicase XPA binds to it, the RNA pole will stop there, and it will activate the XP proteins. So RNA pole also act as a sensor for damaged DNA. And how RNA pole will do it? Because remember, in order for DNA to replicate, RNA pole has to first make an RNA primer. So before starting of DNA replication, RNA pole will have to come in place and make that RNA primer. If there is a damaged DNA around where RNA pole will make an RNA primer, it will sense that damage, and it will activate this exogenous repair mechanism in the cell, and it will fix that first, and then the replication will occur. So it's a post-replicative damage repair. Or in a way you can say pre-replicative. Okay, so that's this part of the slides. Then I will talk more about DNA damage repair in the second set of slides. Okay, thanks.